Tonight, while you sleep, your brain will paralyze every muscle in your body. It will sever your connection to the physical world and force you into a hallucination so vivid that neuroscientists describe it with one word: indistinguishable from waking reality. You will not choose what you experience. You will not be able to stop it. And for approximately 2 hours, you will have absolutely no idea it is happening. From a purely evolutionary standpoint, this should not exist. For 8 hours every night, you are completely defenseless, paralyzed, unconscious, unable to run, unable to fight. You are easy prey for any predator that finds you. The creatures that slept longer should have been eaten first. The genes that demanded unconsciousness should have been erased from the population millions of years ago. And yet every animal on Earth with a brain sleeps. Not just sleeps, but dreams. During REM sleep, your brain does not rest. It becomes, if anything, more electrically active than when you are fully awake. The question neuroscientists have asked for 7 decades is not simply why we sleep, but why the sleeping brain stages a full hallucination every single night. The answer is more unsettling than you think. To understand it, we need to go back to September 1953 in a small laboratory at the University of Chicago. A graduate student named Eugene Azarinsky was conducting a routine study on infant sleep when he noticed something no one had formally described. The eyes of sleeping subjects were moving rapidly, in synchronized bursts beneath their closed eyelids. When he woke subjects during these periods, nearly all of them reported vivid, detailed, narrative dreams. Wake them during the quiet phases, and they reported almost nothing. Azarinsky and his supervisor, Nathaniel Kleitman, published their findings in Science in 1953 and named what they had found rapid eye movement sleep. They had discovered that sleep was not a single uniform state of unconsciousness. It was a cycle, alternating between still phases and an eerie interval of intense internal activity. When Kleitman attached electrodes to sleeping subjects' scalps, the EEG readings during REM looked almost identical to those of a fully awake brain. The body was asleep. The brain was not. This paradox would take 60 more years to fully explain. The first part of the answer came not from psychology, but from plumbing. For decades, neuroscientists were troubled by a basic problem. Unlike every other organ in the body, the brain has no lymphatic system. The lymphatic system is the body's waste disposal network, collecting and clearing the metabolic byproducts produced when cells generate energy. Your liver, your kidneys, your muscles, they all use it. Your brain does not. The brain burns approximately 20% of your body's total energy, while accounting for only 2% of your mass. At that rate of metabolic activity, the waste it produces should be catastrophic. So where does the garbage go? In 2013, a neuroscientist named Maiken Nedergaard at the University of Rochester discovered the answer. She and her team published a landmark paper in Science that would fundamentally change how the field understood sleep. The brain, it turns out, has its own private cleaning system. Nedergaard called it the glymphatic system. During sleep, the glial cells surrounding your neurons shrink by approximately 60%. This shrinkage opens microscopic channels between brain cells. Cerebrospinal fluid, the clear liquid that bathes the brain, rushes in through these channels. As it flows, it carries metabolic waste products out. The system works like a pressure flush. The cells compress, then the fluid sweeps everything out. The critical point is this: the glymphatic system only operates at full capacity during sleep. When you are awake, it runs at perhaps 5 to 10% of its sleeping efficiency. Your brain can only clean itself when you are unconscious. When Nedergaard's team analyzed what the system was clearing, they found something alarming. Among the waste products flushed during sleep was beta-amyloid. Beta-amyloid is the protein fragment that accumulates between neurons and forms the plaques that are the defining pathological marker of Alzheimer's disease. The glymphatic system, during a normal night of sleep, clears measurable quantities of it from the brain. A study by Jeffrey Iliff at Oregon Health and Science University in 2014 showed that this clearance was most efficient during slow-wave sleep. A further study, published in Science in 2019, found that a single night of disrupted sleep produced a significant increase in beta-amyloid accumulation in healthy human brains. One night. Tau protein, another molecule associated with neurodegeneration, was also found to be cleared by the glymphatic system. In 2017, researchers at Johns Hopkins found that tau levels in cerebrospinal fluid rose measurably after even one night of sleep deprivation in healthy young adults. These are not proteins that accumulate slowly over decades. They build up every single day as the metabolic cost of consciousness. Sleep is not the absence of productivity. It is the only window in which the brain can pay its own maintenance bill. But the glymphatic system explains why the brain must sleep. It does not explain why the sleeping brain must hallucinate. For that, we need a different discipline entirely. In 2000, a Finnish neuroscientist named Antti Revonsuo at the University of Turku published a paper that proposed one of the most startling theories in modern evolutionary psychology. He called it the threat simulation theory of dreaming. Revonsuo's argument was direct. Dreams are not random noise from an idle brain. They are a Darwinian training program. The sleeping brain uses the safety of unconsciousness to run rehearsals of threatening scenarios, preparing the waking organism to survive them. Revonsuo and his colleagues analyzed thousands of dream reports across hundreds of subjects. The results were striking. The single most common theme in human dream content, across cultures and across age groups, was threat. Threats of being chased, of falling, of physical harm, of social rejection, of being trapped. Not random everyday events, but precisely the kinds of scenarios that would have determined survival or death in the ancestral environment. The content of dreams is disproportionately dangerous relative to the actual waking lives of those who dream them. Most people in the modern world are not regularly chased by predators. But in their dreams, they are. This explains something that had long puzzled dream researchers: why nightmares exist at all. From a naive perspective, nightmares seem like a design flaw. Why would a brain spend the night terrorizing itself? Under the threat simulation theory, nightmares are not a bug. They are the system running at full intensity. Antonio Zadra, a dream researcher at the University of Montreal, has spent decades cataloging nightmare content. His research finds that nightmares cluster around a narrow set of themes: pursuit, attack, natural disasters, and physical harm. These are precisely the scenarios that would have been lethal in the environment in which modern humans evolved. Children's nightmares are even more revealing. Before a child has learned to fear car accidents or financial ruin, their nightmares are almost exclusively populated by predators, monsters and large animals hunting them in the dark. Revonsuo argued these are not learned fears, but the default threat library of the human nervous system, preserved over millions of years. For the brain to run these simulations effectively, it faces a mechanical problem. If the body acted out what the dreaming mind was experiencing, the consequences would be catastrophic. A person genuinely fleeing a predator in a dream would thrash, shout, fall from bed, and injure themselves and those around them. The brain solved this with a mechanism called REM atonia. During REM sleep, neurons in the brainstem send continuous inhibitory signals to the motor neurons controlling the body's skeletal muscles. The result is a complete, temporary paralysis of the voluntary muscles. Your arms, your legs, and your torso are locked. Your eyes move, your respiratory muscles function, but the large-scale machinery of your body is immobilized. This discovery was made by Michel Jouvet at the University of Lyon in 1959. Jouvet performed a series of lesion experiments in cats. When he surgically removed the brainstem region responsible for generating REM atonia, the cats began physically acting out their dreams. They would stand, stalk, pounce, and fight, eyes closed, brains deep in REM. They were hunting prey that existed only in their sleeping minds. In humans, this failure mode is called REM sleep behavior disorder. People with RBD physically enact the content of their dreams. They punch, kick, shout, and fall from bed while completely unconscious. Carlos Schenk at the Minnesota Regional Sleep Disorder Center has documented these cases since the 1980s. His patients have broken bones, punched through walls, and attacked their partners, all while dreaming. They are the clearest possible evidence of what REM atonia was designed to prevent: a body fully synchronized with a hallucination it cannot distinguish from reality. The degree to which the sleeping brain commits to its simulation is total. In 1962, psychologist Charles Fisher at Mount Sinai Hospital documented that during REM sleep, the brain generates false sensory data neurologically identical to real sensory input. The visual cortex, the auditory cortex, the somatosensory cortex, all of them are firing. The dreamer is not watching a film. They are generating an entire world, from the ground up, in real time, using the same neural machinery used to perceive actual reality. There is no internal marker that flags the experience as synthetic. The reason you almost never question a dream while you are inside it is not a cognitive failure. It is a feature. The simulation has to feel real to be useful as training. The scale of what happens when this system fails makes the point viscerally clear. In 1964, a 17-year-old named Randy Gardner was supervised by Stanford sleep researcher William Dement as he attempted to break the world record for continuous wakefulness. Gardner stayed awake for 264 hours and 25 minutes, just over 11 days. By day 4, He was experiencing microsleeps he did not know were happening. By day 5, he could no longer perform simple arithmetic. By day 6, he was hallucinating while awake. A street sign had become a person. A store window had filled with a scene from a dream. His brain, denied its nightly hallucination, began hallucinating during the day instead. The simulation did not stop. It leaked. There is a disease that makes this point with lethal finality. Fatal familial insomnia is a prion disease caused by a mutation in the PRNP gene. It strikes adults, typically between the ages of 40 and 60. The first and defining symptom is a progressive inability to sleep. Over the following months, every cognitive function collapses: panic, hallucination, dementia, and death. There are fewer than 200 known cases in all of medical history. Every one ended the same way. Without the nightly glymphatic cleaning cycle, toxic proteins accumulate until the brain can no longer sustain function. No sleep, no survival. So consider what this means for tonight. While you sleep, your brain will simultaneously run a janitorial crew, a survival training program, and a complete sensory hallucination. These are not separate processes happening in parallel. They are a single, integrated system that can only operate when you are not present to observe it. You are maintained by a process you cannot witness while it runs. Think about the last nightmare you had. The racing heart, the desperate running, the absolute conviction that the danger was real. That was not a malfunction. That was your nervous system doing exactly what millions of years of natural selection designed it to do. Running the drill one more time. Running it so that if the threat ever materialized, the response would already be wired in. Think about the last time you held yourself together under pressure. The moment you chose not to panic when every instinct said to. That composure was not something you generated in real time. It was practice, loaded in while you were unconscious. Matthew Walker at the University of California, Berkeley, has spent decades documenting the relationship between REM sleep and emotional regulation. After one full REM cycle, the brain's prefrontal cortex, responsible for rational judgment, comes back online fully recalibrated. After 20 hours of continuous wakefulness, your cognitive performance is equivalent to being legally drunk. The difference between a mind that handles the world clearly and one that comes apart under pressure is often a single night of uninterrupted sleep. There is a line that runs through all of this research. The madness is the maintenance. The hallucination is the housekeeping. What you call sanity, clarity, and rational thought exists only because something irrational runs beneath it every night. You do not sleep so that your brain can rest. You sleep so that your brain can do the one thing it cannot do while you are awake: clean itself, train itself, and rebuild itself from the inside. Waking up is not simply returning to yourself. It is being handed back a version of yourself that was quietly repaired, rehearsed, and renewed in the dark. The necessary madness ran its course. And now,